My name is Erilynn Heinrichsen. I'm a fifth-year biomedical sciences student working with Dr. Gabriel Haddad to study the genetics of obesity and hypoxia using fruit flies. As part of the Socrates program, my teacher partner is Linda Spenlove at Mira Mesa High School, and we teach advanced biology and AP biology to 10th and 11th grade students. One of the main objectives of this lab, the Teenage Mutant Ninja Fly lab, is for students to both reinforce and support their prior knowledge of the central dogma of mutations, and then to be able to take this knowledge a step further in understanding how mutations can be used in current science to discover more about genes. In order to take this step, students have to think outside the box a little bit and apply their prior knowledge to a new situation, and in doing so, improve their metacognitive skills. On to what the lab actually consists of. On the first day of the lab, you start off with a brief introduction. The students then begin work in a handout. They're each in groups of 4, and each student in the group of 4 has their own sequence that they're assigned. So there are 4 different sequences. And in the handout, they work on transcribing and translating their particular sequence from DNA to mRNA and then to protein. Based on the resulting amino acid sequence, they get to match that up with one of 4 envelopes that are on a table, and each of these envelopes contain a different set of instructions for drawing their fruit fly. Each student has a picture of a fruit fly looking somewhat like this one, on which they can draw the phenotype instructed for by their individual set of instructions. Once the students have drawn their fly, perhaps adding their own customizable aspects of it, they compare their fly picture with the rest of their group. And they notice that not all of them are the same. Each one has one factor that's different. Comparing the different amino acid sequences that led to those flies, they can see that each fly had one protein mutated. They can then figure out that since each altered protein correlated with an altered phenotype, then that protein must be responsible for that phenotype. And therefore the gene that codes for that is transcribed and translated into that protein, that must correspond with that phenotype as well. And it is a relatively complex idea, but that's sort of the point, is to challenge them to take what they already know and to apply it to this new situation. Once they've figured out that they can determine which protein codes for each trait, they get the opportunity to identify each of these traits in real fruit flies. So in the lab, I've listed the 4 types of fruit flies that are needed for the lab, and you can order them all from Carolina Biological Supply. And the students basically get a chance to look through microscopes or with magnifying glasses to see if they can identify which fruit flies have a darker body color or white eyes or no wings, or if they're a wild-type fruit fly and they have all the wild-type characteristics. The students really enjoy this part because it's sort of a free-for-all. It's not a really structured part of the lab. It's more, they have a very simple task of identifying which mutation or which fruit fly mutation links up with each sequence. But really the whole point of this part of the lab is for them to observe live organisms. I've tested this lab in classes ranging from extremely independent and motivated students to classes where the students have a harder time getting the concepts and aren't quite as motivated to work through on their own. And in those type of classes, it required a lot more modeling by me, sort of prompting them on what to do. But then they were still able to think outside the box. They were still able to work through all the important aspects of the lab. And often those classes had the most fun with the live fruit flies at the end. So it really was very successful in all the classes in terms of accomplishing the main objectives. This lab is definitely unique in the sense that it's a true collaboration between myself as a graduate student who works in a lab using fruit flies and Lynn Spenlove teaching advanced biology and AP biology. And it also, very importantly, it also really brings in the fruit fly as a model organism, and it allows them to see that you can learn a lot in science from a fruit fly. And this is something I really want students to take out of this lab, is not memorizing anything in particular, but to reinforce that genotype does relate to phenotype, and that is not just something that we learn in a textbook, but that we actually use every day in the laboratory in science, and is how in science we discover new genes and their importance in diseases and their importance in our health as humans, and that we need model organisms such as fruit flies to get this basic knowledge about our diseases. And so I hope that students really take away, this knowledge that science is cool, science is incredibly relevant and incredibly important, and what they're learning is much more than what they need for a test. And of course that they have fun with it too, because fruit flies are pretty cool.